So for urinary histology, we have three slides for you: the kidney, the ureter, and the urinary bladder, and we have a couple models. Here's an aspect of one of those models. And if you recall from lecture, when we talk about the kidney in particular, we're really thinking about the functional unit, or the nephron. And so you recall from lecture that there's two general regions of the kidney. There's the outer cortex region, and the cortex region, one of the distinguishing features is the presence of the renal corpuscle, which is going to be composed of both the capsule as well as the glomerulus, or the capillary bed. You're also going to be finding the tubules, the both proximal tubule as well as the distal tubule. These are the proximal and distal convoluted tubules. Those are going to be present out in the cortex. Whereas down in the center part of the kidney, you're going to have the renal pyramids, and these are going to contain primarily the big collecting ducts, as well as then the loop of Henle. And for lab, we're going to have you distinguish between the thick segment of the loop of Henle and the thick segment of the loop of Henle. So as we look through these various histology slides, I'm going to try to point out some of those defining features of these different parts of the tubule. And so this first slide here, this is going to be of the cortex, and we know that because of the presence of the renal corpuscles. Again, the corpuscles here, these are the combination of both the Bowman's, or the glomerular capsule, as well as then the capillary bed, or the glomerulus. And so from this magnification, we can see maybe a dozen or so of these renal corpuscles, and we can see here's the glomerulus, the capillary bed, within then the capsule. We also should be seeing some of the distal and proximal convoluted tubules, and a higher magnification will be able to distinguish between those. And so this next slide here, this is a higher magnification showing you these three components of the nephron. So if we look at the tubules first, remember the most common tubule in the cortex is going to be the proximal convoluted tubule. And that's going to be the one here that has the very prominent brush border, or microvilli, that creates almost a closed, very fuzzy lumen. And so these are simple cuboidal cells. You'll notice that the nuclei tend to be centrally located, and they have very apparent microvilli creating this fuzzy lumen. Whereas the distal convoluted tubules, they're going to typically have fewer microvilli, a very minimal brush border, if any, and the consequence of that is they have a much more apparent open lumen in these histological sections. Also, you'll notice the nuclei tend to be pushed more out towards the apical surface. These tend to be less common, so the distal convoluted tubule is going to have the more apparent lumen without the brush border, and they tend to have nuclei push more to the surface. And so again, when you look at the cortex, you should be able to distinguish between the proximal convoluted tubule with the brush border, with the microvilli, and then the distal convoluted tubule without. Here we can see a nice example, there's two of them, of the renal corpuscle. And again, the corpuscle is the combination of the capillary bed, or the glomerulus, as well as then the Bowman's, or glomerular capsule. And so recall that the capsule is going to have both a visceral and a parietal layer. And so this out here, this simple squamous layer right here, this is going to be the parietal layer of the Bowman or glomerular capsule, whereas the visceral layer is going to be directly in contact with the capillary bed. Now, we are not asking you to distinguish between the capillaries, and more specifically the endothelial cells of those capillaries, and the visceral layer. We're going to see an example of a model here in the next slide, and that's the location where you should be able to distinguish between the podocytes, or the visceral part of the capsule, and the endothelial cells. So in these microscope slides, you should be able to identify the glomerulus, that's the capillary bed in the middle. There's also this capsular space, so the space between the capillary bed as well as the visceral, and then this outer parietal layer, this is going to be this capsular space. Now this next figure is going to be one of the models you have in lab. And in addition to the features that we can see on the previous slide, there are a few additional ones that are only going to be present on the model. So here we can see some of the vessels that supply blood to this region. And so more specifically, coming in right here, this is going to be the afferent arteriole. This is the one that's a little bit larger and has more smooth muscle, which is going to allow us then to have better control over the pressure of blood in the glomerulus. So blood's coming in through the afferent arteriole. It's going to move through the capillary bed, or the glomerulus. Remember, these are fenestrated capillaries, so they're pretty porous, but not as porous as the sinusoidal ones we saw in the liver. And blood eventually is going to leave this glomerula via the efferent arteriole. Now, in addition to the endothelial cells that you can see on this side of the glomerulus, remember there is that visceral layer of the capsule. And that visceral layer is made up of these specialized cells referred to as podocytes. And these are those cells that have these feet-like or toe-like projections that are going to wrap around the endothelial cells. And more importantly, you can see that they're not continuous. There's these very, very prominent slits or filtration slits that you can see between these adjacent podocytes. And so on this model, and there's another model, although I think this one's a lot better, you should be able to identify the podocytes. In this case, they're the yellow cells, the filtration slits, which is the little space between the adjacent cells, and then the glomerulus, or the actual capillary bed. In addition to that, you will also be able to see this structure referred to as the juxtaglomerular apparatus. So recall from lecture that this apparatus is composed of two structures. There's the macula densa, that's this collection of cells here that are part of the distal convoluted tubule. And then there's also these specialized smooth muscle cells of the arteriole here, and these are the juxtaglomerular cells. So the juxtaglomerular cells, which are part of the arteriole, and the macula densa, which is part of the distal convoluted tubule, collectively, this area right here is referred to as the juxtaglomerular apparatus. And that's that population of cells that can detect changes in blood pressure as well as solute concentration and respond in a way that changes our blood pressure. Now, this next slide is going to show you what I think is a very good example of that juxtaglomerular apparatus. And so here you can see this is that classic distal convoluted tubule, right? Very prominent lumen without the brush border. The nuclei tend to be pushed more to the apical surface. And you'll notice that this distal convoluted tubule is running right next to where the arterioles would be entering or exiting that glomerulus. And when you see this position, you'll want to take a look at the distal convoluted tubule to see these kind of tightly packed, more columnar cells. And this represents the macula densa. And so although you're not asked to identify the juxtaglomerular cells on the slides, you'll only have to do that on the model, you should be able to identify the macula densa, which again is part of the distal convoluted tubule. Here we can also just remind us that most of these here are the proximal convoluted tubule, the ones that have the closed up, very fuzzy lumen because of that prominent brush border. This next slide here, this is now moving down into the medulla, and we know that because there are no glomeruli. We see primarily tubes here, no glomeruli, and these tubes therefore would be collecting ducts or the loop of Henle, both the thin and the thick segments. And so if we look closely at this is a much higher magnification, we can see two of those three regions very clearly. Probably the most prominent and unique cells are going to be the collecting duct cells. These have these beautiful, simple cuboidal ducts with nice circular lumen, and more importantly, the really circular spherical nuclei and nice separation, nice divisions between the cells. And so when you see these nice cuboidal cells with really, really, really nice separation between cells, that's the classic example of those collecting ducts. As you can see, about maybe 6 of them in this field of view. The other tubes here we can see, these represent that simple squamous layer. This is the thin segment of the loop of Henle. So remember that loop of Henle is that part of the tubule that dips down into the medulla, and part of it is going to be the thin segment, and that's going to be defined by this simple squamous. And so here we can see maybe a few cells, that's all it is, of these simple squamous that make up the wall of these thin segments of the loop of Henle. So all of these are cut in cross-section. Now, this next view is a little different. It's going to show the same area, but it shows us the thick segment of the loop of Henle. So here we can see some nice collecting ducts. Again, nice prominent lumen with really nice cuboidal cells with a nice central prominent nucleus. And again, very nice separation or division between these cells. So here we can see a few collecting ducts, in this case cut in longitudinal section. The other cells that you can see here, these ones that have a similar simple cuboidal look, but have very grainy, fuzzy, almost indistinguishable cells, these are the thick segments of the loop of Henle. And so these become really prominent, especially in comparison to the nice, distinct cells that you see in the collecting ducts. So the thick segment of the loop of Henle, these are simple cuboidal, but they have very grainy and almost indistinguishable cells that make up the boundary, whereas the collecting ducts have also the simple cuboidal, but really prominent nucleus, as well as then distinguished cells. And so within the medulla, you should be seeing thick and thin segments of the loop of Henle, as well as the collecting duct, whereas up in the cortex, you should see the Bowman's capsule with the glomerulus making up the renal corpuscle, as well as then the distal and proximal convoluted tubules. The last structures that we have are the ureter and the urinary bladder. And there's going to be some similarities between them, most importantly, the epithelial lining. So this first one here, this is going to be the ureter. This is the tube that carries the urine from the kidneys to the bladder. And the thing to notice here is we can see the entire circular ureter. We can see the lumen here. It's a pretty small tube when you compare it to the size of the bladder. For this particular slide, we're asking you to identify and distinguish between the tunica mucosa, the muscularis, and then the adventitia. And if you look at this, we can see a very prominent epithelial lining. And more importantly, this is where we find that transitional ET. So remember, the transitional ET is that stratified layer that seems to change shape of the cells depending on how extended or filled the various tubes, or in the other case, the bladder are. Outside that is then going to be this lamina propria. So this is going to be the lamina propria of the muscularis. And then we see a pretty significant muscularis layer. This is going to be smooth muscle. Now, what's different about this smooth muscle compared to the digestive system is here we have an actual outer circular motion muscle and an inner longitudinal. So remember, in the digestive system, it was always the circular in the middle, and then the longitudinal was on the outside. In the case of the ureter, it's flipped. The inner layer of smooth muscle is the longitudinal, and the outer is the circular. But other than that, we still have our mucous membrane, which is composed of the ET, in this case, a transitional ET, and a little lamina propria. And then we can see our muscularis layer with the inner longitudinal and outer circular. And outside that, you can see is just a lot of connective tissue. And this is that adventitia. This is this dense irregular that's going to be holding it to the surrounding connective tissue of the body wall. And so here we can see a lot of adipose tissue and other tissues. Really, this is going to be the ureter that's held. This is a retroperitoneal organ. This is going to be embedded in that posterior body wall. A closer view, a much closer view, is really giving you a good example of the transitional epithelium. So again, this is the stratified layer, and because even out to the surface, the cells tend to still retain a more cuboidal shape, we would call this transitional as opposed to, let's say, a stratified squamous. And so this, you shouldn't misidentify a stratified squamous like you'd see in the skin or maybe in the oral cavity or in the esophagus. This is going to be that transitional epithelial tissue. And then below it would be the lamina propria. This last slide here, this is the urinary bladder. And the difficult part here is at high magnification, this is going to be difficult to distinguish between the ureter. But at this magnification, this is at 40x, we can see the size difference. Right here, we're just seeing one little part of the lumen. We don't see the entire continuous bladder, given how big it is in comparison to the small ureters. It happens to be collapsed in this case, so all of this here is due to the collapsed nature of the bladder in this slide prep. If we were to look at this at higher magnification, you would see the transitional epithelial lining, followed by a little lamina propria. More importantly, you see this really prominent smooth muscle layer, and that's actually several layers, both longitudinal and circular, that kind of blend together. And this has a special name. This is the detrusor muscle, and this is the name for the smooth muscle of the bladder. And so again, this is multiple layers of smooth muscle, kind of like we see in other areas. We have a longitudinal and a circular and then another longitudinal. But collectively, this has a special name, again, that detrusor muscle, which is the smooth muscle of the bladder. And so that's, for the bladder, all you need to be able to identify is the mucosa with the transitional epithelium and then the smooth muscle layer down here. And again, in comparison, this slide with this slide, it's just the sheer size of the two. Here we can see the entire kind of star-shaped ureter, whereas in the bladder, because it's so big, we're not going to be able to see the entire lumen in one particular field of